you very much. And now we have Alana Lecher, Associate Professor at Leeds University.
Thank you, Sarah and Natalie, for organizing this, and also for making it so that I could attend to this wonderful hybrid opportunity. So this presentation really centers around a series of experiments in which various collaborators from a lot of different fields, including geoscientists, biologists, and archaeologists, are seeking to understand and find an answer to the question of what happens to pre-Columbian artifacts when sea level rises and those artifacts are submerged, which we've seen from several presentations today that is going to happen, and we wanted to know what happens when that's the case. So my background is actually in hydrology and biogeochemistry, and I work with archaeologists in Florida to understand how sea level rise impacts archaeological sites, and this is a natural extension of that kind of general research that I've been working on. So I want to give you a little bit of background on hydrology really quick. This comes from a paper that I published with April Watson, who's the archaeologist at Everglades National Park. And we wanted to look at how sea level rise would cause groundwater levels to rise, and then that would infringe and submerge and wet sites. So if you look at this screen here, this would be like a barrier island, for example, the ocean, some sort of buried midden or other cultural resource or site. We have the coastal aquifer, in which there's groundwater and it's saturated. You can also have water that is pulled up through capillary action above the groundwater levels and actually wet the sites above it. So for this little demonstration, first I'm going to remove that capillary fringe label to make it a little bit easier to see that as ocean levels rise, that actually causes local groundwater, coastal groundwater levels to rise as well, through reduced draining of groundwater. And then also on barrier islands, the groundwater system essentially sits on top of sea level that runs through the island, which we call like a freshwater lens. So this means that sites throughout the state of Florida on the coast are getting wet and submerged in groundwater long before they will be overtopped or submerged by ocean water. And we wanted to look at what happens when these artifacts are submerged or wetted by that groundwater. So first, I'm going to introduce the concept of diagenesis, which you may or may not be familiar with based on your background. In geology, where I come from, diagenesis just describes the process by which sediment turns into sedimentary rock. But it's also been applied in archaeology to describe chemical, biological, and other physical changes that occur to artifacts after they're deposited and buried. And you can see from this figure from Wilson and Pollard how different ways that diagenesis can occur with artifacts, which is represented by the bones. And you'll see that there's actually 3 different types of water in this figure: soil solution, soil water, and groundwater, all of which mediate and can accelerate diagenesis, which diagenesis can remineralize and strengthen artifacts, but we've actually found it can go the other way as well, and often does, and degrade artifacts. So I'm using artifacts from this location in Boca Raton. It's South Inlet Park. It's on a barrier island, so very much area that is subject to those forces I showed you before of rising groundwater levels due to rising sea level. And we did 2 experiments focused both on shell and bone artifacts. So this is animal bone. The shell we collected from a shell midden, and then we used isotopes with our collaborator from the Smithsonian to assess the impact on the shells. And then for bones, we collected from a buried site that essentially had like a very dense bone layer, which seemed to be some sort of food processing site. And we used a series of wetting experiments to understand how diagenesis was occurring in the bones. So I'm going to talk about the isotopes first, but I want to kind of say that, you know, when Dr. Watson and I published that paper, we had like 4 different viewers all asking us the question of what happens to artifacts after they're submerged in water. And while there's a generalized framework, which kind of comes from the forensic science literature, biological sciences, there's not really been a lot of research actually on demonstrating what actually happens on artifacts, especially in the Southeast US. There have been some experiments done in Europe, but that's mostly been relegated to either metal artifacts, which we don't find metal pre-Columbian artifacts in Florida, or on animals and fauna that we don't have in Florida, so such as sheep. So for the isotopes, a little bit of an isotope review is that like basically each element that we have, such as carbon or nitrogen, if you can add an extra neutron, you can get a different weight of that isotope, and that's what we use to study are these kind of anomalous isotopes that you typically don't find in nature. We use a ratio of the common isotope to the uncommon isotope, and we normalize it to a standard to just basically get a number that tells us some sort of ratio of how this isotope, how abundant it is in that sample compared to everything else in the world. Isotopes can be really useful because they can tell you things like if that animal is a carnivore, an omnivore, if it's an herbivore, are its food sources marine or terrestrial. Then it can also tell you things about like environmental changes. If you look on this plot, the x-axis is carbon, C13, which is the isotope of carbon we use, and the y-axis is nitrogen. And these different animals plot on different locations on the graph based on their food sources. And there's been some research done in California and the Pacific using shells and shelled middens to try to look at determine changes in oceanographic conditions throughout time, kind of using the middens, the history of the past, and then trying to link that to changes in what was happening with the cultures at the time. So like were there changes in their food sources that caused changes in how the people adapted to that and that change. So our study, we looked at 3 different types of oyster, or actually 2 types of oyster and clam. These are the first time that these studies have been looked at in an archaeological context, these specific species, using isotopes. And we looked at modern oysters and donax, and we compared them to artifact donax that we excavated from the midden. So I want to show you just some quick results. So this is the depth of the midden. We did 3 different depths within the midden. And we'll look at the N15 first. So this is nitrogen 15, the uncommon isotope. You can see that we have the oyster and the clam, but they seem to have the same pattern with depth, which looks like this is a very well preserved in the archaeological shell, and we can use that to infer changes about the environment. However, the C13, the carbon isotope we use, it looks like it's kind of all over the place. There's no consistent change with depth. So something else is affecting and changing the chemical structure of the shell in this midden. What we think is happening, and this has been shown at other archaeological sites, although it hasn't been published, it's all in review or in process of current, is that the amino acids within the shell are basically breaking down at an uneven rate. So amino acids are like the building blocks of protein, and we need 13 different amino acids basically to build protein. And this plot shows how the carbon and nitrogen ratio changes with the C13 value. Basically, it's a fancy way of saying that the amino acids that have a lot of carbon are breaking down relatively quickly. That's making it so that we can't actually use these shells to study past climate because the sea level rise is basically destroying their chemical signature. On to bone diagenesis. Bones have natural porosity to allow for fluid movement. However, when bones are exposed to water, that can also degrade them by dissolving them, breaking down different parts of the bone. And then as the bone dissolves, its porosity increases, allows for more water to be absorbed. And again, this is something that's been mostly studied in Europe and not Florida. We used bone wetting experiments where we basically took different types of bone based on different taxonomy, location, and purpose in the body, and age or depth within the site. We weighed it, submerged it in water for 24 hours, dried the exterior to make sure we're only measuring how much water was absorbed, and then weighed it again. The idea being the more water a bone artifact can absorb, the greater the porosity and the more diagenesis has occurred. This is a big figure. I'm just going to give you the highlights. This is our shallower sample at the top and our deeper at the bottom. And then we have the different species or bone types across the bottom. So whether it's species, a family, an order, etc. And what we found broadly is that vertebrae tend to be of any type, tend to be very prone to diagenesis and destruction. So it seems like there's more of a loss of vertebra than of other bone types, even accounting for different types of animals. And then fish spines and fish scales are relatively well preserved. So it seems like fish artifacts are well preserved, whereas vertebrae seem to be really degraded through diagenesis, and we're losing them. And that brings me to my conclusion, which is basically just a summary of everything I've said. And I hope to be, or we're planning on submitting this to a journal article soon. So hopefully you'll have more time then to peruse and see the different differences for each of the animal types we looked at. Thank you.